folks, welcome to another set of Chemistry 2 video notes. In your second lesson of our biochemistry unit, we'll be talking about nucleic acids. By the end of this lesson, you should be able to describe the structure of the two types of nucleic acids, describe the processes of DNA replication, RNA transcription, and RNA translation, and explain how those processes relate to protein synthesis. So let's get started. It might have been a while since you've studied biology, so let's do a little review on the structure of the cell, the basic building block of life. There are many different types of cells that perform different functions within a living organism. But for all of their variety, they have many features in common. A typical animal cell is shown here. All cells are enclosed in a cell membrane. Within that membrane is a gelatinous fluid called cytoplasm, which suspends a number of smaller cellular components called organelles. Among those organelles are the nucleus, which contain the genetic material that determines the cell's function, lysosomes, which contain enzymes that break large molecules into smaller molecules, the mitochondria, where those smaller molecules get oxidized, providing energy to the cell, the endoplasmic reticulum with its ribosomes, where proteins are made, and centrosomes, which play a role in copying chromosomes. In this lesson, we'll be particularly focused on events that happen in the nucleus and in the ribosomes of a cell. In the last lesson, I mentioned that there are 4 classes of biomolecules. We talked about proteins then. Today, we're talking about nucleic acids. In the next video, we'll talk about carbohydrates. In our last video about biochemistry, we'll talk about lipids. Nucleic acids are a class of biomolecules that are responsible for carrying genetic information and regulating the formation of proteins within a cell. There are 2 types of nucleic acids, DNA and RNA. DNA stands for deoxyribonucleic acid. It's found in the nucleus of a cell and has a molar mass on the scale of 1 million to 1 billion grams per mole. RNA stands for ribonucleic acid, which is found in the cytoplasm of a cell and has a molar mass on the scale of 25,000 to 1 million grams per mole. Both types of nucleic acids are a type of biological polymer, made up of monomer units that are called nucleotides. A nucleotide consists of one sugar molecule bonded to both a phosphate group and an organic nitrogen-containing base. In the picture on the top right here, you'll see a sugar in green, with a nitrogen-containing base in light blue, and a phosphate in yellow. Both of the last two attach directly to the sugar. Each nucleotide links up with other nucleotides with its sugar and phosphate parts, leaving the bases to be bonded as side chains on the sugar. In the picture here on the bottom, you can see the sugar-phosphate backbones for both RNA and DNA, and how those nucleotides link together. Those 2 backbones probably look very similar to you. It might take you a minute to study those pictures and even see a difference. The difference is that sugar that makes up each nucleotide. Let's look at those sugars a little closer. Ribose sugar is found in the nucleotides of RNA, so we call those nucleotides ribonucleotides. Deoxyribose sugar is found in the nucleotides of DNA, so we call those nucleotides deoxyribonucleotides. The difference between ribose and deoxyribose is highlighted here in yellow. On carbon number 2 of a ribose molecule, there's an OH group attached. If we trade the OH out for just hydrogen, removing the oxygen altogether from that second carbon, we call it deoxyribose. That's the only difference between those 2 backbones. Sugars are a class of carbohydrates, which we'll talk about in the next lesson. So for now, that's all you need to know about deoxyribose and ribose sugar. The phosphate groups for both DNA and RNA are exactly the same, but the nucleotides of DNA and RNA have a different selection of bases. Here are the 5 bases, each of them an organic molecule containing nitrogen within their cyclic structures. Both DNA and RNA use 4 bases, 3 of them being adenine, guanine, and cytosine. The difference is in the 4th base. In DNA, we use thymine, but in RNA, we use uracil. The bases are a really important part of nucleic acids. All of the genetic information that determines the type of cell and how it operates in the body is coded within the sequence of those bases in nucleic acids. That said, it's extremely important that the sequence of bases, therefore the genetic information, be maintained when we create new cells for growth and repair, or when we create proteins. In order for that to happen, there are several processes that occur, but we'll focus on just the three main processes of replication, transcription, and translation. DNA replication happens within the nucleus and is the process of copying DNA to make new DNA molecules, which happens during cell division. Transcription also occurs in the nucleus, and it's the process of creating various types of RNA molecules using a DNA molecule as a template. The RNA is then transported out of the nucleus into the cytoplasm. Translation occurs in the cytoplasm, more specifically in the ribosomes, and it's the process of using the RNA to create protein molecules by linking amino acids in a particular order. So let's talk about these steps in more detail. To talk about DNA replication, you first have to understand the structure of DNA. We mentioned before that nucleotides link together to create a sugar phosphate backbone with bases attached as side chains. There are actually two of these backbones in a DNA molecule. The bases pair up together in the middle, while the sugar phosphate backbones stay on the outside. Because of the shape of each of those nucleotides, the way the backbone is held together, and the hydrogen bonding that occurs between bases on either side of the DNA molecule, DNA takes the shape of a twisted ladder, which we call a double helix. Imagine it as a rope ladder that's all twisted up. The sides of the ladder would be the backbones of the molecule, the sugar and phosphate bits, and the rungs of the ladder would be the base pairs held together by hydrogen bonds. Those bases pair up in a very specific way. Adenine only ever pairs with thymine, and guanine only ever pairs with cytosine. In case you've forgotten what hydrogen bonding is, it's a special type of really strong dipole-dipole attraction between molecules. It only occurs in places where a hydrogen is attached to a nitrogen, oxygen, or fluorine. This is due to the huge difference in electronegativity between hydrogen and those three elements. This electronegativity difference causes a partial negative charge on the nitrogen, oxygen, or fluorine, and a partial positive charge on the hydrogen. That partial positive charge on the hydrogen will attract itself to a nitrogen, oxygen, or fluorine atom on another molecule, because those atoms usually have a high degree of negativity. And as you know, positives and negatives attract. At this point, you're probably wondering, why the heck is he telling us all of this, and why does hydrogen bonding matter right now? That's a great question. Look at these molecular structures. Adenine has one of these partial positive hydrogens right next to another atom, nitrogen, that is partially negative. Thymine's exactly the same way, except it has oxygen instead of nitrogen as its partially negative part. These two molecules fit together like a puzzle, with two places of hydrogen bonding occurring. Guanine and cytosine are in a similar boat, but with three places of hydrogen bonding. These regions of hydrogen bonding are exactly why adenine only pairs with thymine and guanine only pairs with cytosine. Now that you understand how bases pair up, let's talk about DNA replication. To start off DNA replication, we unravel our double helix. In other words, that hydrogen bonding between bases gets overcome by another force, and those two backbones get split apart. This allows each side of the DNA strand to now act as a template for creating a new strand. Those bases from the original strand are now left out in the open and are able to hydrogen bond with the bases of a new nucleotide. The nucleotides that come in pair up exactly with the bases they're supposed to pair with. When these new deoxyribonucleotides attach to the old bases, we form a new sugar phosphate backbone and an exact replica of our original DNA strand. You can see this process pictured above. The green backbones are from our original DNA, and the yellow backbones represent the new ones that are being formed. If we read the DNA sequence of a section of this DNA, we might see CAGTGC. In the original DNA molecule, the opposing strand was sequenced with its complementary base pairs in the same order. So C would go with G, A went with T, G went with C, T went with A, G went with C, and C went with G, giving us the complementary base pair of GTCACG. When the new nucleotides come to pair up on that same section of the DNA, they'll also be the same complementary bases, creating an exact replica of our original DNA strand. DNA replication doesn't have anything to do with creating new proteins, but RNA transcription does. In RNA transcription, we create strands of messenger RNA, or mRNA, that take sections of the genetic code from the DNA to the ribosome so that they can make new proteins. The processes of RNA transcription and DNA replication are exactly the same with one exception: uracil. Instead of a new DNA backbone being made with deoxyribonucleotides, we create a new messenger RNA backbone using our ribonucleotides, which include one of the 4 bases: adenine, cytosine, guanine, and in this case, uracil. Adenine will still pair up with thymine, and cytosine and guanine are exactly the same as they are in DNA, but this time if we have an adenine in our DNA strand template, it'll be paired up with its complementary uracil instead of thymine. Take this picture here, for example. Our original DNA strand had AATC-AGT as its base sequence. If it were just replicating, its complementary base sequence would be TTAG-TCA. But since this is RNA transcription, the complementary will have uracil instead of thymine, giving us UUAGUCA. You'll be asked to come up with complementary base sequences for homework, so make sure you understand this bit. You may want to copy down the chart on the top right here. The order in which those bases are lined up, otherwise known as the base sequence, is extremely important. It determines all of our inherited traits and plays a major role in the creation of proteins, which is what we're really here to talk about today. That protein synthesis happens through RNA translation, which we'll talk about in a second. But first, let's crack that genetic code. If you remember, all proteins in the human body are made up of some combination of some of the 20 amino acids. The base sequence of RNA holds the code for the order in which those amino acids are bonded in a protein. There are only 4 bases, but there's 20 amino acids. So if each base coded for 1 amino acid, we can only have 4 amino acids, and that just doesn't work. If we were to use 2 bases together to code for our amino acids, that gives us 16 different combinations, still 4 short of how many amino acids we have. Therefore, the order in which 3 bases are linked together, giving us 64 different possible combinations, gives us our code for our amino acid. That 3-base sequence is called a codon. Each of these 64 possible codons corresponds with an amino acid, or it's a signal to stop protein synthesis. So for example, the sequence AUG is the codon that signals the start of protein synthesis, as well as corresponding to the amino acid methionine. On this chart, you can see which codons respond to which amino acids. That information is also included on that chart of amino acids I gave you in the last set of notes. Some amino acids, like methionine or tryptophan, only have 1 codon. Others, like leucine or serine, respond to 6 different codons. This chart is useful if you want to create a base sequence that'll code for a particular sequence of amino acids in a polypeptide. The opposite of that chart is the codon wheel. It helps you decode a sequence of bases to figure out what amino acids will go in what order in the polypeptide chain. We'll go back to this chart in a little bit so you can see how to use it. Alright, so we have the messenger RNA that carries a version of the DNA sequence to the ribosomes, but we've got to get the amino acids to the ribosomes as well. That's where transfer RNA, or tRNA, comes in handy. A transfer RNA molecule consists of about 75 ribonucleotides in a strand that bends back on itself and creates 2 regions of double helix shape with a big twist in the middle. This creates what looks like an upside-down L-shaped molecule. At the closed loop at the bottom of this molecule is a 3-base sequence that is complementary to the mRNA molecule's codon, which we call an anticodon. This anticodon corresponds with a certain amino acid, which is attached to the opened end of the tRNA chain on the top right of the molecule. This sets up our tRNA molecules to carry amino acids from one part of the cell to the ribosomes, where translation takes place. Here we are, RNA translation. Our messenger RNA has taken the message from the DNA's genetic code, transcribed into RNA language, and is now processed by a ribosome. As it's being processed, each codon in that mRNA gets paired up with its complementary anticodon in a tRNA molecule. As this happens, the transfer RNA molecule carries over an amino acid, which then forms a peptide bond with the previous amino acid that was carried over. This creates a polypeptide with a particular sequence of amino acids, which, as you know, is simply a protein. So now that you know the whole process in which proteins are formed, let's go back and look at that codon wheel. So to use that codon wheel, we need to understand a little bit about the way we write our DNA sequences. The DNA strand has two ends, the 3' end and the 5' end. When we write the base sequence for a section of DNA molecule, we often write it from 3' to 5' going left to right. For any homework you do for me, you'll be given a DNA sequence in that fashion. As you just learned, we don't go straight from DNA to proteins. We've got to transcribe it to RNA first, and then translate that to decode it into amino acid sequences. So you need to find the complementary RNA-based sequence for your DNA strand. In this example, we're starting with a DNA sequence of TACAAGCTGGTACCTATC. Remember, when translating to RNA, an A pairs with U, T pairs with A, and C and G pair together. So the mRNA sequence would be AUGUUCGACCAU Notice on the DNA strand, we have the 3' end on the left, but on the mRNA strand, we have the 5' end on the left. If you're given a problem that starts with mRNA, you'll have the 5' end on the left, which is where you want it. Now we can use that codon wheel to translate this into a sequence of amino acids. First, read from left to right and find the first occurrence of AUG. That's the only codon that signals the start of protein synthesis. Once you find AUG, split up your codons from there. Remember, a codon is 3 bases together. Next, pull out your codon wheel, and we'll see which amino acid each of our codons corresponds with. Our first codon is AUG, so go to the middle where the 5' is and start with A. From there, go to the next level and find U. Then, go to the next level after that and find G, and that'll tell us that our first amino acid is methionine. Next, we have UUC. So start with U in the middle, go to U on the next level, and then go to C. That codon codes for phenylalanine. Keep doing this for the rest of the codons until you get a codon that tells you to stop. When you finish decoding all of your amino acids, write out the polypeptide chain as we did in our last notes. For this one, you get a sequence of amino acids that looks like this. Well, that's it for today, folks. Hopefully you were able to learn what you were supposed to learn. As always, if you have any questions, please ask.